So in my lab, we're interested in how cells and tissues communicate with each other so they know how to differentiate properly. So I'm interested in how the communication happens, both from cell to cell, but also within the cell, how that happens. And usually what's happening is that you have some sort of signal transduction mechanism inside the cell, which eventually changes some activity of transcription factors that make their way into the nucleus and turn on different target genes depending on what cell you're talking about. So I'm very interested in how the communication happens from cell to cell, within the cell, and also gene regulation. So my lab works with fruit flies, and I'll get to why that's so important in just a second. We do a lot of microscopy, and we also do mathematical modeling of these systems. And we also do cloning. So there's a lot of genetic manipulation of flies. That's why flies are so important, is because they are easy to manipulate genetically if we really want to dissect what's happening at the level of the gene regulation. For the microscopy, the cloning work is important for either using CRISPR or traditional transgenesis techniques to tag certain proteins with GFP so that we can use live imaging, live microscopy, to track the protein concentration and also protein-protein interactions in live tissues and embryos over time. I'm very interested in the quantitative aspect of these particular processes, so being able to turn our microscopy images into something like XY data or XY time data for protein concentrations. And using the fruit fly is really important for getting that level of quantification of the protein concentrations. The goal eventually will be able to use the modeling work in a predictive way to be able to control some of these medically relevant signal transduction pathways. Usually the feedback that I give is in the context of the way that they've explained the work that they've done. And that might be something that's corrective, like, okay, well, this is a good work that you've done. I think it might be helpful if you tried this next. Whether it's positive feedback or critical feedback, it's usually wrapped up into that kind of discussion. I don't have any specific expectations for work hours, like what time to get in during the day, how long to stay before you leave for the night at home. I don't try to push that on my students, and they understand that I do expect a certain level of productivity. It's really up to the student also to be self-motivated. Self-motivation is one of the top qualities that's going to make a successful graduate student. In my lab, we have a pretty good track record of having students get into either an academic type job, so like going on to do a postdoc, or into some sort of industry job. The skills in my lab prepare you for a variety of different job options in the end. So usually this comes towards the end of your PhD when we're talking about different options. I advise a student who wants to do a postdoc, like, how do you go about getting a postdoc? Or contacting some industry contacts, people that I've known, either graduate students that I've known or advised in the past who are in industry positions, seeing what kind of openings are out there. Also, I am open to having students do like summer or semester-long internships